Hello everyone. So today we will be doing mitosis in onion root tip. First of all, the aim of this experiment is to study the different stages of mitosis in onion root tip, or we can say to prepare a temporary mount of onion root tip to study mitosis. Okay, now the requirements. So first of all, we'll take a medium sized bulb of onion and we will trim off the old roots from its base by means of a sharp blade. After that, we'll place the onion on a conical flask, or we can use the glass bottle also. It should be full of water and you can see with its roots touching the water, and we'll keep it for a week to see the roots, and we will finally find that after a week the roots has grown up like this. We can see here in a week the roots has grown to its maximum length. Let's see what all other things we are required here for doing this experiment. So first of all, this compound microscope, that onion bulb with grown root tips, glycerine, methanol, acetic acid, hydrochloric acid, petri dishes, forceps, pipette, dropper, needle, brushes, watch glass, staining, cytocarmine, burner, we can use candle also instead of that, matchstick, scissor, the beaker, flask, blotting paper, pipette, slides box, cover slip. Now the procedure. First of all, we have to prepare a fixative. For the preparation of fixative, we have to take acetic acid and methanol. We'll take a beaker also where we'll keep the acetic acid and methanol with the help of pipette. The ratio is very important. We have to keep the ratio 1:3. Acetic acid 1, ratio methanol 3. Means if we will take 1 milliliter of acetic acid, the methanol should be 3 milliliter. So for preparation of fixative, you can see we are taking 1 milliliter of acetic acid with the help of pipette. We are using 10 milliliter marked graduated pipette so that we can properly and very precisely take 1 milliliter of acetic acid into this beaker. After that, we'll clean this pipette and we'll take 3 milliliter of methanol. Very precisely, we will take 3 milliliter of methanol just to keep the ratio 1:3. Acetic acid and methanol should be in the ratio 1:3, and like this, our first step to prepare fixative is completed. Okay, after that we'll take the onion bulb with the grown root tip here. We'll cut 3 to 5 millimeter of the tips of root and we will put it in the petri dish. After that, we'll take the mixture of acetic acid and methanol into the another petri dish. This is the fixative which we have earlier only we have prepared it. After that, with the help of forceps, we'll transfer the root tips into the petri dish containing the fixative. Fixative we have prepared. It is a mixture of acetic acid and methanol in the ratio 1:3. So we'll transfer all the root tips from the petri dish to another petri dish containing the fixative. Now we'll keep it for 1 hour. For an approx 1 hour, we'll keep our root tips in this fixative medium in this petri dish containing the fixative. So this process is called fixation. Approx after 1 hour, we'll take out these onion roots from the fixative solution and we'll wash it with the help of distilled water. After that, we'll take some hydrochloric acid with the help of dropper into the other petri dish. After taking out the HCl solution in that petri dish, we'll transfer one by one the onion roots with the help of forceps here to the petri dish containing the hydrochloric acid solution. For this step, for our convenience, we have transferred the hydrochloric acid along with the onion root tip into this watch glass. As you can see here, we have to heat it up to 60 degrees Celsius for 15 minutes. Now the idea behind this, that we have to hydrolyze the root tips by warming them to about 60 degrees Celsius in one normal hydrochloric acid solution. So after 15 minutes, what we will do, we'll take out these onion root tips from the hydrochloric acid solution and we'll use the distilled water to wash it. Again we will wash it thoroughly. We have taken another petri dish here. After that, you can see we have taken one staining solution, that is acetocarmine. Now we will take one another watch glass here and we'll put some acetocarmine. As you can see, we are taking acetocarmine into this watch glass. After that, what we will do, we'll again transfer those onion root tips with the help of forceps into this watch glass containing the acetocarmine solution. Now, after transferring all these onion root tips here, what we will do, we'll again keep this watch glass containing acetocarmine staining solution along with that onion root tip into this flame. You can see we have to heat it gently so that stain अच्छे से properly it could get into it. Okay, after that you can see here we have got very properly and nicely stained root tips. We'll take glass slide, cover slip, needle, we'll keep a blade also here, blotting paper. We'll keep our slide very neatly. Okay, with the help of forceps, we'll take out one root tip here. After that, with the help of blade, we'll cut a very small section of this root tip. We are taking very small section of root tip here, and the extra root tip we'll discard it with the help of needle. Now we'll place in the middle this root tip we have placed in the middle, and we'll try to keep our slide very neat and clean. So extra stain we have cleaned it. Now we'll place a drop of glycerine on this slide. So we have put 1 drop of glycerine over the hydrolyzed root tip. After that, we'll place a cover slip on the root. Very gently, we'll gently squeeze the root by tapping the cover slip with the blunt end of a pencil or a needle until the cell separates and spread out into a very thin layer. Okay, so like this we can tap it and we can press it very gently. And one thing we should must assure that our slide should be very neat. Okay, now you can see our slide is ready to be observed under the microscope. So let's see it under the compound microscope. Now the observation part. We'll observe the slide under this compound microscope. So we have placed our slide properly over there. Now we have to adjust it. We have to focus it properly. We'll set the mirror properly here. After that, first we will observe it under the low power of the microscope to locate the dividing cells. And after that, what we will do, we'll examine the different stages of mitosis under the high power of microscope. So finally, you will be able to see here the different stages of mitosis. One by one, the interphase, prophase, metaphase, anaphase, and lastly the telophase. Okay, so let me discuss one by one very briefly. So under the low power of microscope, rectangular cells with pink nucleus are seen, and under high power of microscope, following stages become distinct. First, interphase. It is a non-dividing phase of the cell cycle between two successive cell divisions. Chromatin fibers appears in the form of a network within the nucleus. Nuclear envelope and nucleolus are distinct. Second, prophase. Chromatin materials shortens and condenses into thread-like structure called chromosomes. Each chromosome consists of two chromatids joined at a point called centromere. Nuclear membrane and nucleolus start disintegration and disappear at the end of prophase. Third, metaphase. A bipolar spindle develops in the cell. Chromosomes become arranged at equator of the spindle. Each chromosome get attached to the spindle fiber at its centromere. And the most basic thing you should know, identical part of this metaphase, that they form a metaphasic plate at the equator. Fourth, anaphase. The two sister chromatids of each chromosome separate from the centromere and move towards the opposite pole. The daughter chromosomes, separated chromatids, appear V, J, L, and I shapes depending upon the position of centromere. Telophase. Fifth, the last stage. The spindle disappears and the daughter chromosomes uncoiled to form chromatin fibers at the two poles. Nuclear membrane, nucleolus reappears. Two daughter nuclei appears at the opposite poles. Cytokinesis occurs by the cell plate formation between the two daughter nuclei. Here we come to the end of our experiment, that was to study different stages of mitosis in the onion root tip. This experiment was performed by Swati Kumari, Ranjit Kumar, Ritesh Kumar, Vivek Kumar, my very sincere student of class 12th biology of Radha Govind Sri Secondary Public School, Ramgarh. Special thanks to Professor Amit Kumar. Thank you.